And what I'd like to do today is discuss some of our recent research on the development of an animal model for the detection of hyperacusis, which is quite a challenging piece of work. This is inspired by the work of Salvi University, who's done a lot of work on stress, role of stress and tinnitus. So hyperacusis can be induced in various ways. One of the most common ways is by hearing loss. People that are receive a hearing loss oftentimes develop tinnitus, and another concomitant of that is hyperacusis. This is particularly common among combat personnel to develop hyperacusis because they not only get the hearing loss, but they get chronic stress. So chronic stress has been long implicated in tinnitus and hyperacusis, but there's been very little work done on the development of animal models for this. So this is an example of pretty extreme hyperacusis. This is an actual patient. He has severe hyperacusis. He's a professional. He's a financial advisor that lives in California around the age of 55, and he has severe hyperacusis. One of the ways we can test for loudness hyperacusis is to measure what's called the uncomfortable loudness level. We put patients in a sound booth. We turn up the sound level. When the sound gets up to about 100 dB HL, most normal in hearing individuals will tell you that that's becoming uncomfortably loud. This person, this professional, has uncomfortable loudness levels that are extremely low. At 125 hertz, sounds around 40 dB become uncomfortably loud. At 250 hertz, it's only 50 dB, 500, 55 dB. As you go up to the higher frequencies, his uncomfortable loudness levels go up to 75, and then they fall back to 65. So these are sound levels that are typically encountered in a normal environment. So I've been at meetings with this individual. He usually walks around wearing earphones, and he avoids noisy places. So this person is really highly stressed out by his hyperacusis. For hyperacusis, there's an axis called the hypothalamic pituitary adrenal axis, abbreviated HPA axis. When we're under normal resting conditions, our stress hormones, which in humans is called cortisol, is at a relatively low level. But when we're stressed, those cortisol levels rise. So the cortisol is released from a gland called the adrenal gland. It's released into the bloodstream and flows from the bloodstream throughout the nervous system. And when it reaches two areas of the brain, the hypothalamus and the anterior pituitary, the cortisol basically triggers negative feedback. And these two hormones, the cortical releasing hormone and ACTH, they basically act to stop or blunt the stress response. You don't want to be stressed forever. You want to have a stress response, and then you want to blunt it, because if you were stressed forever, you'd be in terrible physiological condition. So, as I mentioned before, one of the pieces of research that stimulated our research was done by Salvi University, and the name of her study was The Sound of Stress. And they looked at this in people that were suffering from tinnitus, and they studied the stress hormones and the HPA axis. And the way they did this is by inducing what's called psychosocial stress. What they had people do is give a speech, and before they gave the speech, they measured the level of the stress hormones. And then right after they gave the speech, they would measure the stress hormones. Okay. So the stress hormone in humans is cortisol. You can measure that in saliva, you can measure it in the blood, and you can measure it in urine. So this is a study from Salvi University study. It's really quite interesting. On the y-axis over here, they're measuring the cortisol levels. Okay. Before the speech, when the person was preparing the speech, they would measure the baseline levels. Then the individual would start giving the speech. They would measure the cortisol levels then. And right after the person completed the speech, they would measure the cortisol levels. And then they would continue to measure them for 30, 40, 60 minutes afterwards. So a normal individual has a nice stress response. When a person has to get up and give a speech, before they give the speech, their cortisol levels are relatively low. And just as they start giving the speech, the cortisol levels start to rise. By the time the speech ends, they're sort of at a peak, and then the cortisol levels fall down, and about 60 minutes later, they're finally back to baseline. When you test individuals that have tinnitus and hyperacusis, what you see is a blunted stress response. The hypothalamic pituitary adrenal axis has been disrupted. The feedback mechanisms are not working the way they should. So here's a tinnitus patient in the dash line. You can see preparing to the speech, the cortisol levels are low. The speech starts, but the cortisol levels barely rise. And at 40 minutes after they've given the talk, the cortisol levels have gone up just slightly, and then they fall back to baseline. So this study provided very convincing evidence that in patients with tinnitus and hyperacusis, the HP axis was disrupted. So we thought this was a very interesting study. We wanted to see if we could do this in animals. Now, one of the first challenges you have as a scientist trying to measure hyperacusis in animals is you can't ask them what levels of sound are uncomfortably loud. You have to find some behavioral readout that be used to assess the animals. So there's different types of hyperacusis that have been described in the literature by Richard Tyler and a bunch of associates. One is called loudness hyperacusis, just when things are too loud. Another one's called avoidance hyperacusis, where sounds are loud and people who have hyperacusis avoid them. They don't want to be around them. Probably a related one is fear hyperacusis. When you hear a loud sound, it evokes fear. And there's a fourth type of hyperacusis called pain hyperacusis. Today, my studies are going to focus on basically loudness hyperacusis and avoidance hyperacusis. So how can you measure loudness hyperacusis in a rat, in our case? So we food deprive a rat, put him in a sound testing chamber like this, and the rat can basically get some food by put his nose in this nose poke over here, and the rat has to wait for a sound to come, okay? And when the sound comes, the rat pulls its nose out of the hole and goes over here and gets the food. Now, how quickly the animal reacts to the sound and gets the food depends on sound intensity. So we want to measure the reaction time to pull the nose out and go and get the food. So here we have a broadband noise. We present it, and we're waiting for the rat to respond. The rat responds over here with some delay. That's the reaction time. We don't want the animal to behave erratically, so we have what are called catch trials. The animal puts his nose in here, there's no sound, and if he just waits a long time, he'll get a food pellet. So there's a way of controlling false alarms. Over on the right would be pretty typical data from a rat model. Here we're measuring the reaction time to get the food. On the x-axis, we're measuring sound pressure level. And you can see that at low sound pressure levels, 30 dB. The reaction times are around 215, 220 milliseconds. And what happens as the sound level goes up, you see the reaction times get shorter, 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 and shorter. All of us are aware of this. When somebody yells at you very loudly, you respond quickly. If somebody speaks to you quietly, tells you to stop, your response is much slower. So rats behave in a pretty predictable manner when they're well trained. To accomplish something like this takes about 6 weeks of training to get the animal up and running to do something like this. So we have a way of measuring loudness perception. We had done a lot of work on a drug that's long been known to induce tinnitus. It's called aspirin, and the active ingredient in aspirin is sodium salicylate. And we know from the clinical literature in humans, as well as animal studies, that if you give a high dose of sodium salicylate, this is going to induce tinnitus. We also know from the clinical literature that hyperacusis is often accompanies tinnitus. So if you have tinnitus, there's a good chance that you might have hyperacusis. Nobody had ever actually tested this with sodium salicylate. So we thought, why don't we see if animals that develop tinnitus also develop hyperacusis? So what we did is we took some rats, we trained this on the reaction time intensity function, and then we measured their reaction time before we gave them the drug. Those are the blue symbols over here, round. You can see the reaction times start off long. As we increase the intensity, reaction times get shorter. Very nice, predictable results. If we take the rats and we give them a high dose of sodium salicylate, about 300 milligrams per kilogram, which we know will induce tinnitus, we see a change in this reaction time intensity function. At low sound levels, the reaction times have actually become louder, implying that things are less loud for the rat. And one of the reasons we think this occurs is this drug, salicylate, causes about a 20 dB hearing loss. So when the animal's listening to this, it's just barely above the threshold for hearing. But as the sound level goes up to 40, 50, 60, 70, and 80, you can see there's a rapid decline in reaction times. The rats respond faster. They catch up to the normal reaction time, and then they become super fast. They're faster than normal, between 50 and 90 dB SPL. So this is pretty convincing evidence that salicylate not only induces hyperacusis, tinnitus, but also induces hyperacusis. This is the first demonstration of something like this. So we said, geez, what's going on with these rats? Are they being stressed out by the high dose of salicylate? So what we did, we took the rats, we gave them various doses of salicylate. 0 is the control, 50 milligrams per kilogram, 150 milligrams per kilogram, or 250 milligrams per kilogram. And we took a blood sample, and we didn't measure cortisol. In rats, the stress hormone's got a different name. It's called corticosterone. And we measured how much corticosterone was in the blood. So in the basal resting conditions, the cortisol levels are pretty low. If we give a rat only 50 milligrams of sodium salicylate, this is a pretty low dose, doesn't induce tinnitus, and the cortisol levels remain low. But as soon as we get up to 150, 250, or higher doses of salicylate, the stress hormones really spike up and they become quite high. What's interesting about these graphs is 150, 250, or higher doses of sodium salicylate. These are exactly the doses that are needed to induce tinnitus with a high dose of sodium salicylate. So this is one of the first pharmacological inductions of hyperacusis with the drug. Another technique we wanted to do was to see if rats would avoid loud sounds. In other words, measuring avoidance hyperacusis or sound avoidance. We had to develop a new way of measuring this. Nobody had ever done this before. And we read through the literature, and there's a technique where you can put rats in a dark box or give them an opportunity to be in a bright open space. Rodents, like rats and mice, prefer to be in a dark location. They don't want to be out into the open because they're afraid a predator is going to swoop down and eat them, like a hawk or some other predator. So if you have, we have a apparatus over here that's a dark box, has a runway leading out of the dark box and goes over here to a bright open arena. And we can put the rats in here and see what their preference is. Do they prefer staying in the dark box, or do they prefer staying into the open arena? So over here shows the response, the amount of time they spend in the dark box when it's quiet. When the sound, this box over here has a speaker in it, we can put sounds into this, and presumably if the sounds are loud enough, they will drive the rat out of the box. So under normal resting conditions, when there's no sound, the rat spends about 95% of the time in the dark box. When we put 60 dB of sound into the box, the rat doesn't like that, and he spends less time in the dark box, drops down to about 75%. And when it gets really loud, 90 dB, it only spends about 60% time in the box. So now we have a way of measuring sound avoidance. What we wanted to see is if we could induce sound avoidance hyperacusis and loudness hyperacusis by stressing the rats out. And the way we did this, we fed the rats in their drinking water corticosterone, and we had them drink the water with the corticosterone for 28 days. And then we ran our behavioral tests of hyperacusis. We also ran some electrophysiological studies to see what was going on in the central nervous system. So here we're measuring the loudness hyperacusis. We're measuring reaction time versus sound intensity. Before we give the animals the corticosterone, stress hormone, we measure the reaction time. Those are the blue open circles over here. You can see the reaction time start off long, they decrease, and they get extremely short at 90 dB SPL. After we give the animals the corticosterone for about 3 or 4 weeks, and we measure the reaction time, you can see the reaction times are much, much shorter. They're much shorter at all intensities. So our interpretation of this data is just chronic stress hormone administration is enough to induce hyperacusis. Let's see if we can convince ourselves that the stress hormone is really doing something. So one of the ways we can do this is look at avoidance hyperacusis, where we have the dark box and the open arena. Remember, the rats want to spend most of the time in the dark box. They only will go out of the dark box if we make a loud sound. And this shows the response of the rats prior to giving them the corticosterone in blue over here. So under quiet conditions, the rats spend about 95% time in the dark box. As we increase the sound level, they move out of the dark box into the open arena. We can drive them over there with the sound. After we put them on corticosterone and treat them, you can see they spend less time in the dark box at 60 dB, and there's a huge difference at 90 dB. The difference at 90 dB is statistically significant. So we've got 2 measures right now of hyperacusis. One, loudness hyperacusis, where we measure reaction times, and we can see that the rats really don't find it much louder, and they also avoid it when we put them in the light dark box. So one of the questions we had is, what's actually going on in the peripheral nervous system and the central nervous system? So one of the things we did with the rats after we put them on this corticosterone regimen for a long period of time is we took a control group and we measured the responses from the inner ear. We wanted to see if corticosterone would disrupt the response from the inner ear. So we measure a potential called the compound action potential, the CAP, and we measured that as a function of sound intensity. And the blue open circles show data from a control group that didn't get the drug treatment. The filled red circles come from rats that were given 28 days of corticosterone. And as you can see here, the data, there's absolutely no change in the response from the cochlea. This is the neural output of the cochlea, the gross neural output. So this data comes from 8 kilohertz tones, but we get the same results at all different frequencies. So no effect. Many of you are familiar with the auditory brainstem response. For that, we put electrodes on the surface of the skull. We can measure the far field response that comes from the brainstem. And we found no change in the auditory brainstem response either. What we did see is a change in the responses that came from the auditory cortex. To do this, we take an electrode and we plant it on the auditory cortex of the rat. We get nice baseline measurements, which are shown over here in the black line. You can see the response occurs later in time. The peak response occurs around 10 to 15 milliseconds, the area shown in gray over here. This is a response farther up the auditory pathway. So the black line shows you the response from a normal, untreated rat. And if we take the rat now and we feed it corticosterone for 28 days and we measure the auditory cortex response, you can see there's a huge increase in the response. So the auditory cortex has become hyperactive. We don't see any signs of hyperactivity from the cochlea. We don't see any hyperactivity in the brainstem, but we do see hyperactivity as we go further up the central nervous system. So we wanted to see if we could measure what was going on with the hypothalamic pituitary axis. So in Salvi University's study, they had psychosocial stress where they had people give a speech. We can't have the rats give a speech, so we had to devise some other technique for measuring the stress response. Well, one of the ways you can do this with a rat is you can take a rat and simply put them in a small container, and when they're forced to stay in this small container for a long time, they find it very stressful. So before we start the study, we get a baseline measure of cortisol. So the rats, before they go in, we measure the cortisol levels, or corticosterone levels, not cortisol. These are the control rats that are unstressed. These are the rats that have been given the corticosterone for 28 days. Their corticosterone levels are actually a little bit lower than the control groups, but there's no significant difference over here. Then we take and we stress the rats out by putting them in this cone over here for about 45 minutes. Then afterwards, as soon as we take them out, we get a blood sample and we measure the corticosterone in the blood. These are the control rats that were untreated by the drug. You can see they have a strong stress response. The corticosterone in the blood really skyrockets. But in the animals that had been given corticosterone for 28 days, you see a blunted stress response. The corticosterone levels go up, but the rise is much less than in controls. This is almost exactly the same with finding that Salvi University found in her tinnitus and hyperacusis patients. So what else is going on in the brain? Well, one of the things we can do is measure a receptor for corticosterone called glucocorticoid receptors. So we know that the auditory cortex becomes hyperactive. So we take out the brain of the rats that have been treated and the control rats, and we can take sections of the brain. This is a section from a control rat in panel A over here on the left. And this is a section from the corticosterone treated rats over here. And we have an antibody that recognizes the glucocorticoid receptors. And just looking at the panel on the left and the right, you can see these dark spots over here. They're much more intense and much more prolific in the auditory cortex of the rat that's gotten corticosterone. You can measure the density and number of these black dots in the control rats in green, and you can measure it in different layers of the cortex. This is layer 6, and this is another layer over here, 2, 3. So these are layer 6 in the control animal. This is the corticosterone treated animal. You can see in layer 6 over here, the much darker dots. This is layer 2, 3 in the control. This is layer 2, 3 in the corticosterone treated rats. And you can see much darker spots. So we go in under a microscope, we measure the relative optical density of controls and the corticosterone treated animals. You can see the corticosterone treated animals have much more pronounced labeling of glucocorticoid receptor in layer 6 and also in layer 2, 3. So we see some biological basis for this hyperacusis. We see hyperactivity in the auditory cortex, and in the auditory cortex, there's an upregulation of the glucocorticoid receptor. So what we think is going on in hyperacusis is sounds come into an ear, they're processed at the level of the cochlea, and in animals that have hyperacusis, we don't see any evidence of hyperactivity in the peripheral auditory system. But as you go up the auditory pathway, you go up to the auditory cortex, the responses from the auditory cortex are much larger than normal. What we think is going on is the brain has a mechanism for turning up its volume control to make things sound much louder. And this increase in gain is what we think causes hyperacusis. This is a model that seems like it fits quite well with things like noise-induced hyperacusis and other forms of hyperacusis that have been measured in animal models and also in humans. So I'd like to stop there, show you some of the preliminary work we've done on developing models of hyperacusis in animals. I'd like to thank my colleagues, in particular, Senthilvil and Manohar, who actually executed most of the studies over here, along with Dr. Chen, Dr. Auerbach, Dr. Adler, and Kelly Radzowan. This research was supported by the National Institutes of Health, the Deafness and Communicative Disorders Subdivision, as well as the Hearing Health Foundation. So I'll stop here and entertain some questions afterwards. Thank you.